Welcome everyone to Wednesday night at the lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Cooperative Extension. And on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday night at the lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Beth Planalp. She works at the Wasteman Center here on campus. She was born in Cincinnati, Ohio, and that's where she went to high school. The high school is called Wyoming High School. It's easier than going to Wyoming to go to high school. Then she got her undergraduate degree at the University of Notre Dame in biology, and then got her master's degree at Loyola in Baltimore in clinical psychology. And she went back to the University of Notre Dame to get her PhD in developmental psychology. And then she came here to UW-Madison in 2014. Tonight, she gets to talk to us about one of my favorite topics, the emotional world of infants. Who thought it would be so applicable to the adult world? So those of you who are grandparents, or who want to be grandparents, this is always going to be an interesting thing to relive some of your fondest memories. Please join me in welcoming Beth Planalp to Wednesday night at the lab.
So my name is Beth Planalp. Thank you, Tom, for introducing me so nicely. I will be talking about babies today because everyone loves babies and everyone loves watching baby videos, so hopefully you will all enjoy that as well. My research broadly focuses on emotion in infancy and how they learn to express emotions, recognize emotions, and regulate their emotions as well. And more importantly, as we go through the talk, you'll see what changes how kids learn how to do that. What changes how infants learn how to understand what emotion means and what it is within kind of the broader context of the world they live in. So to start us off, because I've heard that this is an engaged group, I have a little activity planned, actually. So the first 30 seconds or so, I'm going to go through a series of pictures pretty quickly. Just yell out the emotion that you think you're seeing in the photo. It's all faces, so it shouldn't be too difficult, but just say out loud what you think the emotion that you're seeing is, okay? We'll go a little fast. Okay.
Happy, angry, scared, surprise,
Scared, surprised, it's actually the same thing, and we'll go over that. But so, you kind of all said happy or sad or angry, and everyone said similar things if you were paying attention. There were some differences a little bit, but in general, you all recognized the same types of emotions, which means that maybe emotions are universal, maybe they're just definitional variations on different kinds of emotions, but in general, you all saw the same thing, which means that emotion isn't new to anyone. You all see it, you all think it. It's not a new topic, it's not something hot kind of in the media, it's something that people have thought about for a very long time. Emotions motivate our behaviors and our thoughts, so everyone understands what emotion is, which actually makes it really, really hard to study because people think they know exactly what you want them to say or what you want to hear, which makes it harder to study emotions in people specifically. But people have been doing it for many, many, many years. So Greek, theorists, philosophers, Hippocrates, and then Galen, several hundred years later, used to study emotion as temperaments. And they thought that the body's humors, so blood, phlegm, yellow and black bile, were all described people's tendency to behave in certain ways. So for example, I don't know if this will show up in the video, but a choleric person here, the person on the left here, ooh, okay, well, that's good to know, can be kind of difficult and fussy. Sanguine person, the next person is a little more happy and excitable. The next one, it's hard to see on these things. Melancholy, so sad, and then phlegmatic, which is difficult or angry. So these were identified in 300 BC or so, or kind of started to be talked about in 300 BC, and then with Galen in 120 AD. So for 2,000 years, people have recognized that there are different ways of behaving and identified those kind of based on someone's emotion or mood that they tend to be in. This continued with Darwin. So Charles Darwin, who we've all heard with evolutionary theory, in 1972 wrote a book called The Expression of Emotions in Man and Animals. And he thought that emotions, this is actually probably the first book or textbook kind of on emotion that we still use in science. I have it on my bookshelf at home, actually. But that emotions are universal. They cross species. They are maybe applied differently in humans, and they are more evolutionarily adapted in humans. But he saw that there were common universal emotions across different species. Which brings us to more modern takes on what is emotion. So there are two broad thoughts on how we define and think about emotion. The first one is more dimensional. So it's across two dimensions of kind of identification. So the first is low arousal and high arousal, right across here. So if you're high reactive and you kind of feel very, very excitable, or low reactive, so I heard someone say bored for one of the cat, right? The cat was bored. It was you. So that would be kind of a low arousal emotion. And then again, or also across valence, so positive and negative. So for example, if you're really happy and you're excited to be experiencing something, then you would maybe fall in this corner up here. If you're relaxed and you're kind of going with the flow and calm and contented, you have low arousal, but you're still pretty positive. So that would be in this over here. If you're sad, low arousal, so you're not really kind of anxious about something, but it's a little negative. So this is a dimensional model of emotion, or also called a circumplex model. You can see it's kind of shaped like a circle. The other way of thinking about emotions is more categorical or discrete, and maybe you recognize this. This is from the movie Inside Out that came out 4 years ago. It's a Disney Pixar movie where we have discrete named emotions, the ones that you all called out earlier. So sadness, joy, fear, disgust, anger. And actually, the original theory was that there were 6 emotions and surprise. So Paul Ekman is a researcher in the 1960s, 70s. He came up with this kind of definition of what are the basic emotions and identified these 6 emotions as basic. So Paul Ekman was actually a consultant on this film and helped them develop the characters and identify which emotions to talk about and how to talk about them. And they did not put surprise in the film because they couldn't figure out how to make it different from fear. So they would have 2 characters that were almost exactly the same. So they combined surprise and fear into the one character. But in general, these are more discrete, categorical ways to think about emotion. There are other theories that kind of add in other emotions like guilt and pride and shame. But if you think about it, infants don't feel guilt or pride or shame. There's a social component, a moral judgment component that comes into play with some of those emotions that come online a little later. So they are called, they're not part of the basic emotion process. They are actually in a theory called DET, differential emotions theories by Carol Izzard. So those are, kind of some additional emotions that adults feel, and it's more universal in adults, but not necessarily in infants. Which brings us to, why do we study infants? Like, why, if we can't study all the emotions, do we study them when they're this young? It's because infants haven't figured out how to stop us from seeing them yet, right? They cry and scream and they smile and they giggle and they laugh, so we can really study pure emotion before they've learned how to modify it or inhibit it or regulate it. So infancy is a particularly interesting time, I think, to study emotion because they can't tell me that I'm wrong either necessarily, right? So it makes it very difficult. I have to infer what they're doing and kind of try to see how they're responding to specific stimuli and understand what they're thinking or feeling, but they can't necessarily stop their emotions. So you can really tie into this basic emotion theory by studying infants. Other thing with infants, though, which is both exciting since I'm a developmental psychologist and a little difficult to handle, is that they change a lot and very quickly. Kids learn to walk and talk, and their cognitive processes come online, and they learn to explore their environments. So we study emotion in similar, in ways, but as they get older, kind of have to adapt what our processes are, or how we think about what emotion is, or as they learn to look around the room, what are they doing to help themselves with their emotion and to express their emotion? So emotions are both fun to study in infants, but also difficult because they change so quickly. and they're so context dependent. However, I'm trying not to use too many different words for what I basically mean is the same thing, but temperament, so like the underlying emotional reactivity to something, the way you respond to something, even though the kids are changing and growing and walking, the way they respond is very, very similar to the way they responded 6 months ago and the way they responded 6 months before that. So the underlying tendency to respond to a situation remains the same. So emotions are changeable in their expression, but they're also very enduring over time. Now, to illustrate that, I have 2 videos of children who are no longer infants, but that have very different ways of expressing their emotions. So in the laboratory, we videotape and we code behaviors that infants are doing. We try to use tasks that are similar to what they would experience at home. So in this case, with both of these videos, you see there's an enticing toy, a fun toy, and we don't let them have it. So we are hopefully eliciting some sort of anger or distress response because they want the toy and they can't play with it, which if you have multiple kids, siblings take away toys. Or if kids are sitting in the grocery store, in one of the grocery store push cart things that I'm blanking on the word right now, but they want a toy, they want candy, you don't give it to them. So kids have to go through these processes all the time just in their natural environment, and we try to tap into that in the laboratory as well. So we think these are very ecologically valid ways to measure emotion in children. So for the first video, and I'll start that and then you can hopefully hear it, just the same child at 8 months and then 18 months. And these are both just about a minute, 30 seconds long. Hope if I can push play. Actually, I'll start it right here. Not too bothered. Just watching the toy, trying to grab for the toy, but he's really fine. Same child, 10 months later or so. And we've adapted the task now so that there's a large container that they can't get in, so it's still taking a toy away or not letting them have a toy, just age appropriate for their motor behaviors. Oops. But you can see he's playing with the toy, but not super bothered by the toy, or not being able to have the toy. Okay. So the next video is slightly different. This is a little girl who's kind of frustrated about not being able to have the toy. She wants it, but we're not giving it to her. And 10 months later, and then she just gives up, which I think is great. Yeah. So, but they have similar patterns of responding. So the first child, even though it was 10 months apart and it was a slightly different, I can stop this. This just continues. Yeah. So even though it was 10 months apart, they respond in similar ways. So we used a different toy and a different method of kind of eliciting that behavior because they're older and they can grab things and touch things and their hands have more dexterity. But they respond in similar ways. So emotions are enduring. They kind of the expression might change a little bit and the situation might change, but the underlying way of responding is similar. Okay. Are all emotions created equal? I hope not, because that's what I study, right? So the basic premise of everything I do is that emotions develop in different ways and in different families, and different children respond to things in different ways. So are all emotions created equal? Probably not, maybe not. I thought I would use an example of data that we have here at Wisconsin using twins to show kind of how a positive emotion and a negative emotion might not necessarily be expressed the same way or develop the same way, even within the same family. Okay? So positive versus negative emotions. using a similar tasks to the ones in the videos. So we code positivity and negativity as well. So in this case, we have a mom who's playing like a puppet game with her child, and then a mom who's playing peekaboo with her child. These are, again, things that kids see every day at home, game playing and engaging in positive kind of behaviors. So moms are kind of eliciting a positive response from their child. And we videotape them and record them and then code them later. Similarly, with the anger task, we have a shiny toy that the mom kind of holds the baby's arms down, and then one that's just out of reach, and we code these to reflect a negative emotion, or in this case specifically, it's anger, as an example of negative emotions. But so we have positive emotions and we have negative emotions that we scored in the lab. So twins are a special case where we have a little bit of control over what's happening to these two kids in the home. So twins have a completely 100% shared environment. They live in the same house, they have the same parents, they maybe have the same socioeconomic status because they're coming from the same place. Their parents have the same education level because it's two kids within one family. So we have a high amount of control over this shared environment component. With twins, there are two types of twins, identical twins and fraternal twins. And identical twins share 100% of their genomic DNA, and fraternal twins share roughly 50% of their genomic DNA. And someone in this building would argue with me, I'm sure, about a little bit of that. But in general, for the statistical model we're doing, we assume that identical twins share 100% and fraternal twins share roughly 50%. And then we finally have this unique environment component that we just don't know where things come from. So maybe twins are in different friend groups, or maybe one twin kind of had an experience with a roller coaster that the other twin didn't have necessarily. So then there's this unknown, unique, or a non-shared environmental component. So if we expect, those colors don't show up as well, but if we expect identical twins to share 100% of their DNA and 100% of their shared environment, and then we have this extra kind of component, their unique environment, we would expect the relation between twin 1 in a family and twin 2 in a family to be fairly similar. So it would kind of look like this, as an illustration of like the correlation between within a twin pair. If they're not identical, if they share only 50% of their genomic DNA, their shared environment's the same, but their the genetic input on their behavior is not necessarily the same. So we have one twin who might have expressed a lot of a behavior, and another twin who might express a little bit of a behavior, and then this unknown component here that could fall into shared environment, it could fall into unique environment, but it's not the same as with identical twins. So what we do is we compare correlations, so at the extent to which two variables relate to each other, we compare those for identical twins and fraternal twins separately. So for positive or negative affect, for example, so that we take how one twin acted on those behavior tasks that elicited negativity and how the other twin acted on those tasks, and we compare them to each other. If we expect there to be a strong genetic component, then the pattern we would want to see is like this. Or I'm sorry, it's, oh my goodness, it's like this. It's like this one. Because, I'm totally blanking right now. All right, we're just going to go to the next slide. Negative emotions. So, okay. Yes, negative emotions. So, if there's a strong genetic component, then the identical twin, here, this is helping me, the identical twin with twin 1 and twin 2 would have a strong correlation. And in the fraternal twins, the twin 1 and twin 2 would have roughly half the amount of correlation that the identical twins have. So in this case, that's exactly what we see. There's a strong genetic component for negativity because the twins that are identical are more similar than the twins that are fraternal. Okay? I had to get to the next slide where I had the two twins next to each other again. Okay? So with positivity, if there's a strong genetic component for positivity, we would expect to see the same pattern, right? That's not what we find. Instead, with positive emotionality, we see that the identical twins and the fraternal twins are roughly the same. And in fact, this looks slightly different, but it's actually not. These numbers are really, they're not statistically different from each other. So with negative emotion, there's a strong genetic component, and with positive emotion, there's not. Positive emotions might be more environmentally cued or might be learned somehow, right? We can delve into that a little bit further by using more, strangely enough, understand this better, but more advanced statistics that look at not only, so the previous slides only looked at the genetic correlation. It only made an assumption on about the genetic correlation based on some behaviors that we'd seen. Now we can tease apart different similarities within the families to identify this shared environmental component, as well as how much the unique component or differences between the twins contribute to a behavior. So again, in genetics, the identical twins we assume have a correlation of 1, and the fraternal twins we assume have a correlation of 0.5. They have a completely common, so a correlation of 1 common environment or shared environment, and no correlation in their unique or non-shared environment. Using the same data, the same sample, with the anger variables, we see that there is a strong genetic component. So the genetic component, accounts for a lot of the input on negative behaviors that we're seeing. The non-shared, this dotted line here, actually is not different from 0. So the shared environment is not really contributing anything to these twins' expression of anger that we see at roughly 12 months old. And then the unique non-shared environmental component is accounting for a lot of the variation. That could be different experiences, different moods that day, who fed what, they're 12 months old, so who maybe has to have a diaper changed or what they ate. But the comparison then, if we look at positive affect, is very different. So we see there's no genetic effect or input onto the positive emotionality that we see, but there is a fairly strong component for the shared environment. And then, again, similar large component for the non-shared. But even without looking at the numbers, the pattern of results here shows that there's a strong genetic inheritable effect for negative affect and not for positive affect. Okay? What am I forgetting? This is the slide that I was like, I need a blank slide to think about how we interpret what that might mean. So I didn't put this here, but we actually looked at that positive affect and what it might be related to in the home. So we had moms report on their own positivity and their own negativity and kind of family-wide expressions of good behavior and positivity in their family. And we had dads do the same thing. And the mom positivity and the family-wide expressions of positive behavior related to the infant positive affect. So we can teach our kids how to be positive and how to be happy and how to see, kind of positive side of things. Whereas the negative anger variables are a little bit more genetic. Maybe we can use positivity to help counterbalance some of those negative things that we see. So positive affect can be taught, can be learned by children. And I think that's a really positive, happy thing to say about how we can raise, kids and how we can affect policy and what we should be thinking about in long-term approaches to childcare. Okay, there's one. See, I knew I needed to put this up here. What am I forgetting? Regulation. So that lovely two-factor circumplex model. It has arousal and it has valence on it. But especially in adults, it's really hard to think about emotion without thinking about what we're doing to modify that emotion. So I'm forgetting words and I'm forgetting things and I'm freaking out a little bit up here, and you can tell sometimes, but I'm also sweaty and warm and I'm getting really uncomfortable and I'm trying to hide it as best I can, so I'm regulating what I'm doing, right? Everyone does this all the time. Sometimes it's implicit and we don't even know we're doing it, and sometimes it's explicit, like me waving my hands a little bit right here, just to kind of cool down. But I'm just being very emotive. So there's this regulation component, this third factor on kind of an idea of what is emotion. We can't really define emotion, especially in adults, without thinking about what we're doing to regulate it. So emotion regulation refers to the ability to inhibit, so to stop an emotion, stop a negative emotion, to modify, so to maybe take that surprise and make it a fear variable, or to maintain the occurrence or intensity of an emotion. So if we're happy, we want to stay happy. So you can both upregulate happiness and downregulate negative affect, or they all kind of work interchangeably. But emotion regulation is also a really important thing to study. And it's really cool to study in kids because when they're really young, they can't do it, and then they learn how. So we can watch kind of the developmental change in how kids learn to regulate. I kind of gave some of these examples, but in adults, so regulation can be very explicit. We can choose to regulate our behaviors. You can meditate or go for a run if you're stressed out after work. Those are more healthy, adaptive ways to regulate emotion. You could also choose the alternative and maybe, drink your sorrows away or bury your head in the sand and deny everything. So those are not necessarily adaptive ways ways to regulate emotion. But in adults, this is really purposeful, and in children, it's not. So in little kids, infants especially, they really can't regulate themselves, especially before 3 months of age. So at 3 months of age, they really depend on caregivers, on all of us, to soothe, to pat their back, to get them food, to change their diaper. Those are actually all helping the child learn how to regulate. If they need something, they get fussy, and you come to them. Their fussiness is just their way of telling you that they're uncomfortable. Right? They're not trying to be little annoying things like, in their bassinet or whatever. They actually need something. So by eliciting that behavior from a caregiver, that's when they start to learn to regulate. Because then you come and take care of them and soothe them, and that they know that behavior helps their own fussiness or their own discomfort. As they get older, so 6, 7, 8 months, kids they learn to regulate more effectively on their own. So if you have, children, grandchildren, you watch them like when the first time they suck their thumb for themselves, which is closer to like 2.5, 3 months of age. But that's regulation. So that's a self-soothing behavior. They can use like their fingers to kind of pick at their clothes or kind of like smack their lips. Those are self-soothing behaviors. As they have more control over their core stability and their head and neck, they can turn around and look around the room. But those are all regulatory behaviors that seem like they're very standard motor control behaviors that kids develop, but they also impact negativity and negative affect. And then at 12 months, they can explore and regulate more independently. They can use their environment. So if they're crawling and they're unhappy and there's a toy on the other side of the room, they can crawl and go get it. And that's actually, if they're unhappy, that's a form of regulation as well. So it's not necessarily just emotion regulation, but in infants especially, we talk about it as bodily or self-regulation. Okay. So I have a video of, it's like the middle child, the one that's that's very true. It's like the middle child in my family too, now that I think about it. But a child who is fairly well regulated, and it's the same task, but I'm just kind of going to talk through it a little bit and tell you what I'm seeing that they're doing that's regulatory. So he looks around, he looks to help, he looks for someone to get him the toy. He's banging a little bit, so this is not necessarily the most effective regulation to bang, but he's looking for other things to do. If he looks at the toy, he gets angry, and he wants it. And then he's soliciting help. So he's asking for help. We're not allowed to give it to him, but he thinks that should get him the toy and help down kind of regulate his negativity. Making vocal sounds, kind of moving his arms. So all of this, yes, he's getting negative, but he's also trying to do things to regulate. And the same child, little older, and again, you'll notice he's doing the same things. He's looking to the person next to him to help him regulate. He can't, or help him open the toy, which is regulatory. He's using his teeth, yeah. He asks for help. So he's learned that his caregiver is there to help him. So this is a pattern of behavior that over time, kids learn what they need to do to get help, to get attention. He tries to open it, and he asks for help. So at this age, that's a very regulatory thing to do. It's the same thing we're doing when we go for a run. We know what's going to help us. Or the same thing when you call a friend. That's what they're doing. And it's gone again. And he enjoys the game. Yeah. Oh, then he doesn't get the toy back, and he gets a little angry. But so in each of these cases, first to show kind of enduring emotion, and now to show regulation, these are the things that we look for in the lab to kind of see what they're doing. Okay, so I've defined emotion and talked about what that is and what it means and how it changes, and then kind of defined regulation and talked about what that is and how it changes and what it means. And I tie all of these things together with infants. But I know Tom mentioned I work at the Wasteman Center here, which is an interdisciplinary research center where we can put all of these things together. So at the Wasteman Center, I can't see what's here. So if you haven't heard of it, it's a center that was founded in the late 1960s. It was funded by Harry Wasteman, but it was funded by a Eunice Kennedy Shriver Foundation grant that is ongoing. The NIH we still have two large funding grants from the NIH, an Intellectual and Developmental Disabilities Research Center, and then the Kennedy Shriver Foundation still, that kind of help run the Wasteman Center. In that building, we have medical physics and engineering. In fact, there's an MRI in the building on the first floor. We have psychology. I'm a psychologist. There are, the Wasteman Center is home to, let's see, 60 different kind of faculty labs from 27 different departments around the university. So there's medical physics and engineering, there's psychology, brain and behavioral development kind of ties those two together, and then we have physiology and neuroscience as well. So we have people who collect blood for us and kind of are looking at some stem cells in those, the twins that I talked about, actually. So all of this is combined at the Wasteman Center so we can look at lots of different multiple processes that help us understand emotion better. So this slide from before, so at the same time all of these things are happening behaviorally, the brain is also changing, right? So you can kind of see in some of these pictures, but the brain at around, this is a 3-month-old brain, it's kind of just a blob, and it doesn't look super exciting, but 7-month brain, and then finally, this is actually not 12, but 15 months old, you can kind of see the colors and the differentiation between the different types of brain matter in the brain. So these brains are really differentiating and kind of becoming what's interesting to study, right? So even in this short period of time, what the brain is doing is impacting motor and cognitive control and emotional expression. But we don't really know what that means in infants. In adults, the adult brain has been studied extensively, especially with emotion and emotional reactivity, actually by a lot of people at Wisconsin. With infants, it's a little bit still unknown. However, it shouldn't be, because infancy is, I'm biased, but I think the most important time in like the brain maturation process. So from this birth to, roughly, well, in utero, so roughly 34 weeks or so, brain development begins at 22 weeks, continues through pregnancy, and then at like 34 weeks is when there's this huge, like, they call it the growth spurt, the brain spurt of neurogenesis and synaptogenesis, so kind of the brain fine-tuning itself in utero. And then if you see the bottom here, from birth to 6, 8, 10, 12, 2 months, or 2 years, now it's in years, so 12 months to 2 years of age, based on this kind of schematic, that's when a lot of things are happening. In fact, by the time you're, roughly 2 years of age, and this, like, your brain is 80 to 83% developed, what it's going to be as an adult. So yes, brain development continues through early adulthood, but from birth to 2.5 or so is probably the most rapid increase or rapid growth and changes in the brain. So kids are learning to regulate, and they're learning what their environment can tell them and what parenting behaviors, how that can affect them, at the same time that all these brain changes are happening. So this is the foundation for almost everything else we do, like this age, birth to 2. So it's really important to study babies. Putting that out there again. Okay, so how does the developing brain relate to emotion then? How can we tie these things together? And I will be the first to tell you I'm not a medical physicist. I'm not a neuroimaging specialist. I'm a behaviorist. And I was hired to do all the behavior things for a lot of these studies at the Wasteman Center. I work with a wonderful group of people from all of the different disciplines I told you about. We have some in the room that really focus on neuroimaging and the physics of neuroimaging and how we can scan babies. But that's what we do. We scan babies. And my job is kind of to help make them comfortable and help the parents be comfortable and get them in the scanner. And then someone else kind of takes over, which is lovely because we have a very interdisciplinary team that works together really well. Can't highlight that enough. The Wasteman Center is great for that. But if you think about an MRI, if you ever had an MRI, you know that it's loud and that this is not necessarily what we see. What we want is we want to put a baby in a scanner, we want them to be happy and smiling, and we want this beautiful, beautiful picture. And instead we get this, crying and screaming, because they don't like that. MRIs are loud and they're scary and they like shake a little bit, so it's not necessarily a nice place for babies to be. But we can't sedate kids. Parents don't really like that. It's not really easy to get them to sleep unless we do a lot of different tricks. And it's not a fun place to take a nap because it's so scary and loud. So we do some things in preparation for all of that to help scan babies. We scanned, we're at 149 1-month-olds, and I'll be talking about some of that data here, by using some of these. So first of all, we make we have a prep room where we have a pack and play and a rocking chair, and the moms feed the babies, because if you think about when you go to bed, you want to be full, you don't want to be hungry, and you want to be comfortable. So the moms feed the baby and then changes the baby and rocks the baby to sleep. And this little pack and play that we kind of dim the lights and make the room pretty as comfortable as we can for them, for the moms and the babies to be comfortable. We put them in what we call a super swaddle. It's a vacuum sealed life vest, basically, where we suck the air out and it really swaddles the baby. And especially when they're young, babies love to be swaddled. They love that tightness and closeness. So we swaddle the baby in this MRI-safe vacuum. It's a immobilizer is what it's actually called, but parents don't like that either, so we call it the super swaddle. We have an MRI suite that is, you can kind of see this is actually what it looks like. That's Bucky on the bore or in the scanner about to go in. And we make it really dark. We turn the lights off as much as possible so that it's dark. We move the infant from a dark room to a dark room. We try not to jostle them too much. We actually have a cute little board that we can take in. So we keep it dark. And then finally, again, working with a bunch of people at the Wasteman Center who are wonderful collaborators, we have a lot of little tricks and techniques to make the scanner quieter. So we have this. This is actually a huge piece of rubber and foam that goes inside the big circle part of the MRI machine, and it dampens the sound a little bit. We already talked about the super swaddle. These are little mini earmuffs that kind of stick to the baby. They're especially made for babies. And then we also have, they're not noise-canceling headphones, but they're MRI-specific headphones that we take in with white noise. And they actually are made to, as you go into the scanner, it gets louder and louder. The microphone or the earphones get a little louder and louder too, and we play like rain, so that there's a little bit. They hear the noise increasing slightly and incrementally as they go in, so that it's not like a shock to the system when it's so loud. With all of this in place, it's kind of like a really loud refrigerator instead of a crazy loud MRI machine. So the babies mostly, especially at 1 month, which is when we've been scanning them, sleep right through this for the most part. Some wake up. There's nothing we can do about that. We just start the process over and they go back in. But doing all of that, we've scanned 149 1-month-olds. This is actually over a little while ago. Successfully. So it was 97% of our sample that we got successful scans on, which is pretty good. That doesn't work. So these are actually the images that we get from our study. So we have structural images that show us the gray matter of the brain. So that's these two right here. So if you think about brain development, you have gray matter and white matter. And gray matter in the brain is kind of like where all the things are done. The computations are done. It's like the buildings. Everything happens in the buildings. And then you have the white matter that it's not white on here, obviously, but it's these colorful kind of traffic patterns. And so the white matter is how things move throughout the brain. It's the traffic flow. It's kind of the processing kind of goes between like gray matter buildings or so with the white matter. It's a very oversimplified way of thinking about it. But so we can look at not only volumes of structures, but also how effective the traffic flow is moving. Okay? And doing that, so we relate that to some of the behaviors we see in the lab. So we also have the babies when they're 6 months old come into the lab and do all of the emotion things I discussed, and then all of the regulatory behaviors I talked about. So we code how well they regulate visually, so how much they look around the room, and how well they regulate with motor behaviors. So, kind of the thumb sucking and playing with the table rather than getting a negative expression on their face. All of those are very effective, by the way. So in a separate study, not done here at all, but coding all of these behaviors, negative emotions, and then a regulation behavior every second in these paradigms. If the baby engaged in a regulatory behavior within 3 seconds of that negative expression, it changed they didn't have any more negative expressions, basically. So these behaviors actually do regulate negative affect in infants. Oh, okay. This could be interesting. I don't have words. So what we find, very basically, kind of we're starting to look at this data right now, is that in certain areas of the brain that have been identified as regulatory in adults. So again, we don't exactly know what's happening with infants right now. The first kind of bigger studies of regulation in children don't start till the kids are about 5 years old. So in infants, we're assuming that there are similar processes in adults. So we looked at certain brain regions that are the same, or that are implicated in regulation in adults, and saw that infants who have a larger right parahippocampal gyrus, and the parahippocampal gyrus is really important for memory formation and for learning, so that is important for regulation, have better motor regulation. So at 1 month of age, these infants with a larger right parahippocampal gyrus have better motor regulation. Okay, for visual regulation, it's slightly different, but infants who have a larger left, it's actually left, I think I switched the picture, but a larger left insula have better visual regulation. So the insula is kind of like the traffic cop of the brain. So it's not really that surprising that kids who have a larger insula are better regulated. It could just be that these kids are a little bit more developed at this age. But these are both areas that are seen in adults relating to regulation, and we're seeing it at 1 month old as well. So that's promising, at least for kind of the future. But we don't always know that bigger is better, necessarily. So we also look at more microstructure. So the white matter, the traffic flow, what the neurons are actually doing. We have two ways to think about that. The first one is with neurite dispersion. The second is with neurite density. So if you think about your hands and like you think about dispersion, like spreading your fingers, your hands when you use them like this are not as effective as when you can kind of pincer something, right? So we don't want a lot of dispersion. We want them to be effective and streamlined. So in this case, as neurite dispersion gets bigger, wider, regulation goes down. So not only are certain areas of the brain larger, but they're actually less effective, less streamlined, and that's relating to regulation later. And the same thing for neurite density. Density, we want to be good. We want a lot more neurons to be more effective, right? So we want a higher number of neurons in the kind of space that we identified, and that's relating to higher regulation. So not only is volume in the brain different in relating to regulation, but the microstructure, the refinement of what the brain is actually doing is also relating to regulation in ways that we would hope to find. And these are all in areas that are kind of the same as areas we would find in adults. Okay? So there's what is emotion, what is regulation. Think about it in twins, and we can say that emotions develop differently. And then think about it in the brain and say it exists really, really early, which is, I think, impressive and exciting. But when I started grad school, not all that long ago, but many years ago, I wanted to study kids. I love I want to know how you can grow up in the same family and be very, very different. Because we all have siblings that we're very different from, and we just don't understand why, right? But I thought I could study children, and just study children, and it would be great and fun and exciting, and I forgot completely about this, right? So parenting is really, really important. Most of my work is actually on parenting. I mean, it has been for 15 years, but the first 6 months of grad school, I was really gung-ho on just studying kids. So study of child development is inherently rooted within family systems, or inherently rooted within the environment that the child grows up in. So how does parenting tie into all of this? Kids learn to regulate and express emotion based on repeated interactions with their caregivers, right? You saw the kids in the videos looking to their caregivers for help because they knew they could. So parents are important. Oh, I did say that. So briefly, I just have like one example kind of from each study that I talked about. But in this brain imaging study, we're studying regulation, and we have a lot of measures of maternal stress and depression and anxiety. And one of our kind of very initial findings from a wonderful grad student in our lab found that maternal stress is related to motor regulation. So moms who are more stressed in their home, and the stress variable was about 8 different types of questions, but in general, experienced more stress, had children who regulated less. Now, it could be that these moms aren't modeling regulation for their kids. They have too much going on, so they're not paying attention, or their child is seeing ineffective regulatory behaviors. And moms who exhibited more depression had children who used visual regulation more. So you're thinking, oh, this is great. Moms who are depressed have kids who regulate more. But if they're using visual regulation, they're looking away from their mom. They're looking around the room to try to find the source of their regulation. So they're not using their mom as a source of support. Maybe because their mom isn't when she's at home. She's kind of focused on herself or something else or sad or not paying attention to her child. So parenting really impacts how kids learn to regulate behaviors. And this is from the brain imaging study. So this is all kind of data we're working on here at Wisconsin. If we think about involvement, so parent involvement in the home, just basic changing diapers and feeding and playing games and singing songs. In general, this visual kind of regulatory behavior is a good thing, and it increases over time. They're learning to do it a little bit more. And if you can kind of see, I have two lines here. The blue's behind the red. But on average, kids do this the same with both their moms and dads, which is great. My dad loves when I talk about this because he gets to tell everyone that his daughter is a fathering expert, which that's kind of what I focus on. But when dads are more involved, look how different it is. So when dads are more involved, kids learn to regulate more effectively, much more quickly. So dad involvement in the home is different than mother involvement. Now, the reason I think mother involvement, this actually doesn't change. This is exactly the same. It's just statistically the numbers look a little different, but it's all right around 0. Moms are always engaged in the home. Right? So it's not like moms are doing less, it's just that dads are kind of adding on top of that. So when dads are involved in the home, kids regulate more effectively. Okay, so we talked about what emotions are. How are you feeling today? I'm none of these, but maybe a little bit of all of them. High anxious a little bit right now. Babies are cool. They change, they develop. Emotions and behaviors and everything changes with them, which is fun to watch. And parents are important too. We didn't focus too much on parents here, but I have a lot of that. If you have any questions, parents are important too. But why is all of this in infancy? Why is it to relate to anything later? Why is it important later? 2 brief examples of studies that I've worked on where we study child regulation in different age kids, so older kids, or look at parenting, or look at like a health outcome. We study how parenting and regulation relate to health, specifically BMI. So in one study where we had child BMI when they're 3 years old, so these are 3-year-olds now, they're not 6-month-olds or 8-month-olds. We looked at parenting behaviors and how they related to child BMI, and it doesn't relate. There's no significant relationship. That's why this is a dotted line and not a solid line. Parenting did not directly relate to child health outcomes. But when we include child regulation, and in the case, the way we measured it here was dysregulation, so inability to regulate, then parenting was significantly related to regulation, which was also significantly related to BMI. So parenting and child health outcomes aren't necessarily directly related unless we take into account the child's regulatory behaviors. Okay, so this is with 4-month-olds, 20, 30-month-olds, and 36-month-olds. But they're still pretty young. So what's the next step? Well, let's look at them when they're 5, because of course that's a little bit older. We saw the same pattern though. Negative parenting in this case, relating to child weight at 5 years old, not related at all. Oops, already went ahead somehow. But when you add into this regulation component, the dysregulation, then we have a pathway that takes negative parenting to impacts child regulation, which we saw, and then child regulation impacts weight health. So parenting and regulation are important in the childhood obesity epidemic that kind of we see a lot of right now. In general, that's health outcomes. So child regulation, and this is early, young children before 3 years old relating to health outcomes. It's also related to parent-child relationship quality, which I don't have a lot of examples of. I didn't put any examples of this in here, but just to kind of highlight relationship quality with your parent, it teaches you how to interact with peers and with partners later in life. So this is important. Child regulation relates to academic achievement and academic attainment. If you think about kids who can't sit down and open a book or read a book or sit still, they're not going to do as well in school as their peers who can regulate as effectively. Not because they can't do well in school, but because they just don't sit still quite as easily, I guess. And then finally, and the one I want to hit home, is child and adolescent depression and anxiety. Childhood regulation is directly related to problem behaviors, of both internalizing, so kind of anxious behaviors, and externalizing behaviors, so kind of conduct disorders and being oppositional and being angry all the time. This is, I'm going to try to highlight it a little bit, but so estimates say that I think 12 to 15% of children between the ages of 3 and 17 are diagnosed with some sort of anxiety or depression disorder in their lifetime. That's a lot of kids. Like if you think of how many kids are in the U.S., 15% of them have some sort of adolescent anxiety or depression. It cost the United States, the NIH's last estimate was like 2014, $250 billion a year just for this right here. But if we can teach our kids to regulate, teach them how to be happy, and teach them all the positive things that like you can do really, really early in infancy, we can try to help that a lot earlier than I think people have thought was the case before. So everything I'm doing is in kids, and it's fun, but this is why. Like, this is what the interesting thing is. So we can like curtail these problems as early as possible so they don't happen. Okay? And that's pretty much it, actually. So thank you to everyone who like, you know, all the funders and the schools and the funding resources and all of the people I work with. And that's all. Thank you. Sorry. Kind of ended on a big public health. I was told to stay behind the podium. I think I did well. Yeah. That's a question already.
Divorced parents,
Yeah.
in terms of impacting regulation, can they have the same impact as if they were together?
Yes. So it depends on which way you want to go with that. There's a lot of ways to think about that. So you can think about the marital conflict piece. You can think about it as two separate homes or separated parents or any type of parents. So having one of the studies that I did, actually, the last obesity kind of BMI study was actually with children who are in Head Start. So it was a Head Start focused study. They're 5 years old, entering school. They don't necessarily have a second caregiver. Sometimes it's an aunt, sometimes it's a grandparent or a best friend. It's more having someone else in your life to teach you different things. So you have one parent to teach you one thing and another parent to teach you another thing. So it can be, there's a lot of research out, Ruth Feldman is a researcher. She's at Harvard, but she has multiple different affiliations. But she studies same-sex couples, and that's absolutely the same. So none of this is contingent on it being a mom and dad who live in the same house. It's more about the effort involved. Yeah, and then there's a whole other marital conflict piece, which is different. But teaching constructive conflict, even through divorce, actually teaches your kids how to regulate better and decreases all of those externalizing behaviors. Yes.
When you have the slide with the identical twins and the fraternal twins, do you differentiate gender then as well with the fraternal twins?
So in this way we didn't. In this particular study we didn't. There are slight differences, gender differences in some of the variables that we look at, but these are not two that had gender differences, so I didn't look at that at all. When you study, for example, like fear, there are slight gender differences in fear. Girls express fear a little bit more, whether that's because they actually experience it or because they're socialized to express it, I don't know. But when we're studying, you can have a gendered specific model. These models did not. We don't necessarily see, so with positivity, so I tend to study positivity and parenting and how positivity can help everything. But with the positivity paper, there weren't any necessarily gender differences in overall positivity, but girls were much more facially expressive and boys were much more excited. So there are definitely differences in how kids show these behaviors, but I didn't necessarily talk about that or go over that. Yeah. Yes?
Is there a difference between the first child and the second child of the same parents?
Ooh.
Yeah.
So, birth order, especially in twins, is something we talk about a lot in our lab. So, we randomly order them so that it's not, we don't say it's twin, we call them twin A and B, and typically we kind of try to randomize them based on alphabetical letter or whatever, and then we create a random variable. So, we purposefully don't look at that. However, in twins, the first twin out, the twin A, twin 1, tends to be a little more active, tends to have, be a little more robust. They kind of pushed out, right? So they're the first twin out for the most part. There are technically differences in twin 1 and twin 2. We randomize that to try to kind of get rid of that in our analyses, if that makes sense, because they're not exactly the same. And it could be that that's how parents rate them. So parents talk about them differently. Parents always talk about, this one weighed 5.6 pounds, and this one weighed 5.3 pounds. So that's how they think about them, which is why we use laboratory assessments so often, because we hope they're more unbiased in that way. Yeah. birth order's hard. I always get a birth order question because with twins, we can at least control for age differences. Parents, they learn something on their first kid. And then by the second kid, they treat them, and not only is the child different, but the parent has learned how to parent differently, right? So there are trait-like parenting variables, but there's also state-like or child-specific parenting variables. With twins, we can at least control for that age. They're learning on the same twin at the same time. With siblings, that's not necessarily the case, but I don't have any sibling data. Thank goodness. It would be a little confusing. Yes?
You have the graph of the parts
The brain.
of the brain that were developing from minus 2 weeks and then
Yeah.
through 2 years.
I'm just going to go back because it's complicated.
Is there a gender difference in that brain development?
Oh, I could have. So we do have a paper published out of this study, actually, that shows there is. So boys tend to have larger brain volumes and develop a little more quickly. Girls tend to fine-tune a little more quickly. There is not necessarily a difference in the behaviors associated with that, but the size of the brain is very different. Yes, and we actually have that published out of this data. So even at 1 month of age, these differences kind of exist in boys and girls. And they catch up to each other eventually, kind of. But so initially, there are gender differences in brain development. Yes. Mm-hmm. Yeah.
We just recently went to a lecture by a professor of social genetics. Do you remember his name? Okay, I don't remember his name. He was awesome, but so were you. But so nature versus nurture.
Right.
And so it's all about the genome and what they can predict. And if I understood him right, you could take one cell from an embryo and look at the genome, and they could predict so many things, like, for instance, how much education this person was going to have. Based on, I guess, these algorithms, or so in some ways, he was saying nurture is
not important, not a big deal.
So much from the nature, from genetics. So I don't know if you have a comment or if you remember who this guy is. Or
I don't know. So there is a genomics.
Do you know who that is?
Was it here?
Yeah. No, he talked at Middleton Library, but
No, I don't remember.
yeah.
And did he say education or intelligence?
No, education.
So I don't know the answer to that exactly. I do know that we've talked about. So we have.
Were you there?
Yeah. So epigenetics, the idea that your genes change over time in the environment, the way that your gene expresses. So even though you have the same maybe basic genetics, the way that the proteins are encrypted or kind of work, that changes with your environment. So with the air you breathe and with kind of the experiences you go through, especially if they're traumatic experiences, that changes how your DNA expresses proteins. So I would say I don't know much about genomics or specific one cell kind of DNA stuff. I do know that as some of these twins, we're looking at some of that though. There's studies kind of talking about taking arm cells and kind of using that to. This is all at the Wasteman Center, so just go there and ask. But looking at how we can create various stem cell lines to kind of anticipate how some anxious behaviors might be expressed in the twins, actually, in some of the twins that are now, these were babies, but they're now 18, 20 something years old, and so they're doing that now in some studies. That one paper is definitely out and published with that. But I don't know about saying you have one, you can determine or that anything is deterministic. I would disagree, but that's because I study behavior, and so I think everything that we're doing now is really important.
The professor was Jason Fletcher, and he's from the La Follette School.
I don't know who that is.
UW. That's just
Yeah.
who it was.
Well, people I work with probably know who it is. I don't.
I remember what he said in terms of education of the mother was most predictive as to how far the child was going to be educated.
Oh. That's so right. So is that because it would probably be both. So maybe people have the potential to be, intelligent or educated in some way, but it all depends on how your parents raise you. So how the nurture issue would still be at play there. There's expectations involved. I went to high school, you should go to high school. I went to college, you should go. So there's probably a lot of it. That's interesting. Yes.
I thought it was very interesting the difference between positive and negative.
Oh, I'm sorry, I messed that up explaining it too. But.
Do you think it's based on the fact that like the negative would be because at heart we're animals and that based on survival?
Yeah, so it's evolutionarily adaptive. So anger is protection, and I'm going to protect my young, and fear is I'm going to run away when I'm scared. So these are more evolutionarily adaptive mechanisms that are necessary for survival. Positivity, being happy in your life, you just got to feed your babies. So if positivity and even regulation aren't really necessarily adaptive, then they're going to be more learned behaviors. That doesn't mean we can't leverage that to use it as a tool, kind of to help foster, just positive growth in general, though. But yes, I agree. It's very evolutionarily adaptive. Yes.
What's the importance of friends and neighbors in the development of?
Oh, I don't know. I actually don't know that. Mostly because that would be so complicated to study. That would bring in so many different variables. I'm going to say it's wonderful because I have some very good friends watching right here. But
the reason I'm asking is, so if you hang around a kid one day out of 30 or one day out of 6 months, and you observe what you consider to be good or bad behavior, and you either discourage it or encourage it, does it make any difference?
I don't know. I would guess that depending on the age of the child will matter a lot. A young infant who doesn't necessarily have the ability to remember that, or even up to about 6-ish probably, wouldn't necessarily remember one instance. It's repeated interactions with caregivers and friends and grandparents that kind of help develop these patterns of behavior. As kids get older, they remember that one. So this is where the idea of guilt, shame, and pride come into play. That socially constructed emotion where they remember that what they did was wrong. There was a moral judgment. There was a social cue that they can read now. So that's when the later emotions kind of are important, and where a friend saying something or teaching something, they would remember that. Birth to 5, which is where I focus most of my time, I would say no, that probably is not going to necessarily impact, but I have never studied that or read anything about that. But that's where these, the secondary tier of basic emotions is important. So the Carroll Izard's differential emotions theory, he adds guilt, shame, pride, and interest. So interest is one, actually. So there's 3 negative ones, or I guess pride can be both, but interest is one. So having an interest in something, kids don't have an interest necessarily yet. They don't know that they should, or that they remember that they're interested in something. But so all of those social emotions would probably come into play. And those start around 3. So after that, maybe it would be more impactful. Yes?
Yeah, I was just wondering, when you talk about the development of the brain, you're going from birth, what about in the womb?
So brain development in the womb starts at around 22 to 24 weeks. It's not until about 34 weeks or so that any of the kind of processes that I'm talking about, that fine-tuning kind of starts at 34 weeks. Yeah, that's the one that's on here. So what you're feeding, what you're eating when you're pregnant is impacting kind of baby's brain. So very important. Eat yogurt, not processed meats. Yes.
I'd like to go back to the previous question. Is there any evidence that there's a difference between intelligence and education? And that was an attempt at humor.
I was like, I could answer something and say what I think, but yeah, that's funny. No, of course not. Look at me.
I can think of a few exceptions. Myself, for example.
Yeah, I know. I can, mess up words like the best of them. Anything else? No?
If not, thank you.